Shabna, are you able to hear me?
Yes, sir.
Okay, good. Yeah, I'm going to share my screen now.
No, sir, not yet. Be careful.
Okay, like we need to start now. It is exactly time is 6:30, probably like 1 or 2 minutes, like exactly. So we need to start this session. But we announced it around 6:30. 6:32 is close to around 8 o'clock for sure. So one of our associate is going to join, but he is stuck in a traffic. He will be joining in another 5-10 minutes. Uh, like one more thing. Okay, the reason why we want to do this session is very simple. Okay, thumb rules for this session. So thumb rules is first step is like all of you please join with your first name and last name. That is one thumb rule. And the second one is like, all of you put in a mute. And if you have any questions, use a chat window. You can type in a chat window, or you can use our WhatsApp numbers and you can send whatever the questions you have it. You can send it to our WhatsApp. We are going to address these questions. And also, like, if you have any technical difficulties, you have the numbers here. You can please talk to them regarding that technical issues, first of all. Make sure that this session is make it a more productive. For that case, everybody put in a mute. That should be real. Otherwise, background noise is coming up. These are all the things behind it is. Okay, so good evening, all of you. I'm in US, for me is a good morning. So anyhow, that should be fine. The world is like become like a smaller and smaller and smaller, like a village. So everybody can access all the documents and all the information. So now, uh, this session topic is like statistical methodology in clinical trials and drug development. What exactly the clinical trials and what exactly is why statistics is required? Without statistics, we cannot go further for sure. So that is the reason I want to explain like what exactly this statistical methodology. That too, this session is restricted to only like life sciences students and statistics background. Because why we wanted to make these two things is life sciences means like medical sciences, life sciences like see biotechnology, microbiology, zoology, those students and the statistics students. See, industry is looking for overall growth of a student. I think hope you see this overall growth of a student. That means if you want to enter into clinical trials and the drug development area, you must know certain skills. That's more important is what are the skills is required. One is medical terminology, other one is clinical trials terminology, regulatory information, basic statistics, and programming. So, of course, communication is important. So, these five skills is mandatory, first of all. When going with these five skills is the mandatory, if you are going with a pharmacy student, for example, probably he or she knows about, still students are joining. Okay. No. So uh, they know like medical terminology, they know like clinical trials terminology, and like regulatories, etc. But unfortunately, they are not good at the statistics as well as the programming side. That is the problem. That's the problem. So if you are going with a statistics student, they don't know this medical side. Medical terminology, they don't know this. So that's the reason when you want to enter into corporate world, you must ready to learn lot of technologies, lot of things. That's more very, very important. So that is the reason we want to explain what is the skills is required and what are the methodologies we want to go with that. All these things we can do that. So this is the just a target for this, particularly for these two students now. When you open this one is like when you see. So actually this agenda is 6 o'clock, it starts from this. So of course, I told you like, Dr. Kaveti is on the way. He will be joining very soon for sure. It's me and the closing remarks by, technology evangelist Sudhakar Garu. He is going to talk about like the last 10 minutes about this. And any of you, like I know a lot of professors and lecturers is joined in this session. If you want to say something, definitely just raise your hand and definitely you can unmute it and one after another you can talk. Absolutely. I don't have any issues. Issues. No, going back. See, always see like this. 1 to the power of 365 is 1. 1 to the power 1.01 to the power of 365. 01, that means very small quantity, small amount. To the power of 365 is 37.7. Do something, learn something every day. Every day. Suppose you are learning something today. When you are going to the college, I want to learn something. It's very so doing nothing at all versus a small consistent efforts. Mathematically, this is well proven. 1 to the power of 365 is always 1, and 1.01 to the power of 365 is 37.7. You think about it like continuous learning is the key for your success. Continuous learning. So think about like all this is the basic concept about mathematics because I'm like both statistics and mathematics side. all our sessions, like it may be seminars or maybe webinars or maybe like trainings, there is a huge gap between industry and education. Everybody knows about it. Our sessions is going to reduce a gap between industry and education. So always think about this word: success and excuses never talk together. If you want to get a success, don't give any excuses. If you want to giving any excuses, all like you never get a success. So think about this beautiful quotation: success and excuses never talk together, just like at a railway track. If you want success, don't give any excuses. If you are giving any excuses, you never get a success. So that means, of course, everybody knows about this triangle of success. Triangle of success is knowledge. Ask is attitude, skills, and knowledge. Why I invited Dr. Kovet is a attorney. He has offices in 26 countries. Offices. Is a very famous attorney in US. Why I invited him for this session is lot of times student has lot of problems. So that is the reason, like one famous trustworthy attorney with us. Absolutely, that should be good for the students. That is the reason I invited. Of course, he will join very soon for sure. So like this, here you are able to get the knowledge from your college or something here. The goal setting, time management, reasoning, communication, interpersonal skills is required. Most important nowadays is self-motivation, self-confidence, integrity, honesty, optimism, enthusiasm, cooperation, commitment. You must have that one. You must develop those attitudes first. So for that case, triangle of success ask is the very important, very important. When you go to the next slide, of course, this is my profile, something like this. I'm I'm from India, and I took my citizenship here. I'm staying in US for last 28 28 years. So I want to see another meaning of real happiness is quality education. We are the winners in Medidata, New York, hackathon. So that's the situation here. This much. So most important part is like think about this. Learning gives creativity. You need to learn the subject first. Learning creativity. Creativity leads to thinking. Thinking provides knowledge. Knowledge makes you great. So that means if you miss the learning part, you miss everything. So you need to learn the subject. You need to get a knowledge. That's the more important is. So this is a very good quotation. Like, you need to think about it like this. These are all the things. No, of course, education is the best friend, and educated person is respected everywhere. Education beats the beauty and youth. So going back to our session, without statistics, nobody is going to take a wise decision. If suppose if I want to buy one product, you are comparing with other products, comparison. That's nothing but statistics. You are collecting the data and you are analyzing the data. And finally, you are taking a decision. So that is the reason statistics plays a vital role. Nowadays, you want to believe it, a lot of these colleges, they have like a data science. Because without statistics, there is no data science. Without statistics, because statistics is helps managing your data, analyzing your data. That is the reason you need to think about converting massive amount of data into meaningful information. That meaningful information helps management to take better decisions. See, again, I'm using that word, converting massive amount of data into meaningful information. That meaningful information helps management to take better decision. How we want to do this? Taking the data stored in a database or cloud systems somewhere else. You need to access the data first. Then you need to analyze the data, second part. Third part is you need to visualize the data. That somebody see the data report, something like that. That is the full circle. You need to work on those things. For that case, a person wants to enter into corporate, they need to learn a lot of things. They need to learn lot of things, first of all. So statistics, nowadays, everywhere statistics. If you go to artificial intelligence or machine learning, data science, of course, MIS, everywhere, business intelligence, everywhere statistics is there. Better to know. So we are talking more like clinical trials and drug development. Clinical trials and drug development main business is pharmaceutical main business is they want to invent a new drugs. They want to invent new drugs or new therapies. How they want to invent a new drugs? I want to prove that FDA is asking only three questions. Very first question is it drug is safe? Is it drug is safe or not? That is my first question. Second question is it really effective or not? Second, third question is it better compared to existing drugs in the market? There are so many medicines available in the market. You need to prove that, oh, my drug is better compared to existing drugs in the market. My drug has less side effects compared to existing drugs in the market. You need to prove these things. Mathematically, you need to prove these things. Further case, statistics is there. Without statistics, it's impossible to prove that my drug is safe, my drug is effective, my drug is better compared to existing drugs. Further case, a lot of statistics methods is required. That's what we are going to discuss right now. Of course, this is not, I'm not going too depth on statistics because some pharmacy people are going to upset. So, because that is the basic philosophy here itself, basic philosophy. So that we are going to discuss now, in this situation now. Now we are going to, statistical analysis is utilized by 96% of Fortune 500 companies for data analysis. Like if you are in a pharmaceutical company, you are analyzing patient profiles and patient information. Same person, if you are in financial companies, you are analyzing customer profiles and customer information. Who is a good customer, who is not a good customer, something like that. If you are in a insurance companies, you want to identify insurance companies' main business is they are collecting the premiums and they are processing the claims. How speed they are going to process their claims. These are all everywhere. Suppose transportation companies, they want to transport maximum number of people with a minimum time or minimum cost. Like manufacturing products, they want to manufacture quality products. They want to improve the quality without affecting cost considerations. So everywhere they want to analyze the data. For that case, statistics is required. Even though now you can see like that company is lay off this many people because of, even though AI, artificial intelligence comes in, artificial intelligence is also not going to 100% replace this analysis. 100, because parameters are different parameters we need to pass it. For that case, manual interaction is required. So having with the statistics knowledge is always advantage in market situations. Okay. Now I told you just now, like clinical trials, we need to prove that like my drug is safe and effective, and compared to existing drugs in the market, I need to prove that my drug has less side effects, etc. Everything. How we want to do this? How we want to do this? This is the approach, because people are studying, but you need to know about application side. Particularly for a lot of statistics students are in this meeting, I'm telling like a control chart. Simple control chart is like X bar chart or R chart or P chart or etc. Because application side, I want to prove that my products are, that is central line. Central line is like a quality level or something like that. Then I'm applying a 3 sigma control limits, upper control limit and lower control limit. Control chart is conceived and divided by the Shivot. It is a simple pictorial device for detecting unnatural patterns of variations of data. Apply that knowledge into corporate. You need to take the product information and take these different batches, take the average, apply normal distribution, 3 sigma control limits, control limits. Then apply upper control limit and lower control limit, apply all these points. If any point beyond the upper control limit or lower limit, I'm going to reject that one actually. So you are learning subject there, but you need to apply that knowledge into industry, because in pharmaceutical companies particularly, you need to understand like a protocol and SAP analysis plan and a table shell and standards. So these are all important, particularly for this. For that case, apart from the subject, you need to have all these additional skills. Apart from subject, you need to have all these additional skills. Then only you are able to get these opportunities. Okay. Now going back here. Now, statistical methodology is a very essential. We need to go with because think about certain things. If a doctor is making a wrong diagnosis, patient may die. Right. If a pharmacist is dispensing medicine, same thing is going to be happen. Suppose a clinical trial analyst like me, if I made a wrong conclusion, wrong information, thousands of people are going to be effective. That is the reason ensuring the validity and the reliability of the results. That's very important. That includes like randomization, blinding, sample size determination, and minimize the bias. All these things is required. All these things is required to get in that part actually. These are all the things you need to consider about. One minute. No. So what is randomization? What is blinding? This is single blinding, double blinding, triple blinding, and how to calculate the sample size. Lot of times people consider about sample and population. Sample population means a set of individuals having common characteristic is population. A set of individuals having common characteristic. I'm using that word is again female population. That means all are females. Male population, common characteristic, all are male, something like that. Similarly, when sample size is nothing but a finite subset of population is nothing but sample. A finite subset of population is nothing but sample. The best example is if you want to test a blood results, what you are going to do, you don't want to give the whole blood in your body to lab, just you are giving a pinch of blood, and they are going to analyze your blood, and whatever the results, lab results, it is applies to whole body. That is the sample. So when you are selecting a sample is the very important. That is the reason. Suppose if you want to test with a diabetic or something, they are going to take it fasting, and after 2 hours you are taking something, they're going to take it all these things, because right, what I'm telling you is most important is sample size. More important is how much sample we are going to take it. It's not possible for us to make everything is comes under our basket. So we want to identify the sample size. What is the population? What is parameter? What is single blinding? Blinding is very patient don't know anything about it. Double blinding, investigator and patient don't know all these things. That is because they want to minimize the bias and randomization. Each and every individual have equal chance to enter into sample. For that case, we are applying some lottery system or tipped random number method. So many methods are there. First, you need to understand, I want to make sure that my drug is safe or my drug is effective or something like that. That is the one important part you need to make sure that, like this one. Okay. Now going back, now statistics, it has a big ocean, first of all. In this big ocean, most of the times we are considered like very few things, not with like, I don't want to discuss like what is the response surface methodology, all those things, no. Even I studied in my master's, no. Now, industry point of view, application point of view, what it is required, what it is required. So, clinical research form a reasonable and accurate inferences from collected information and sound decisions to presence of uncertainty. Applying statistical is a key for preventing the errors and bias in clinical research. Okay, so descriptive statistics and inferential statistics. Somebody in a chart window, they are asking like e-certificates or something like that. I am telling you, I am not charging anything. I am not giving any e-certificate. That's first step is because you need certificate or you need knowledge. This is unnecessary because you are deviating the topic into other direction. Okay, I am not giving any e-certificate, first of all. That's pretty much. Otherwise, if I want to give a certificate, you keep on asking our office people. We have a lot of other things to do that. That's the one thing he said. Okay, next. Somebody is asking about what is blinding. So, write down that question. Like, suppose, for example, if you are asking about the blinding means single blinding. Suppose, for example, I have like 100 subjects. The word itself is 100 subject. I don't want to use that word patient. I don't want to use that word patient. It's because only two studies is oncology and HIV. Oncology and HIV, we are taking only patients. Remaining all the studies, we are taking healthy volunteers. In this healthy volunteers, like we want to do all these things. Blinding means even patient don't know, subject don't know whether he is taking a medicine contains active ingredient or placebo. Placebo is a sugar pills, dummy capsule, something like that. So they don't know. That is a single blinding. Suppose, for example, if I am an investigator, I know this medicine is for this purpose. What I am going to do, I may bias. Or somebody is suffering with disease, probably I want to give this medicine to them, something like that. So that means bias comes into the picture. That is the reason even I investigator also don't know which whether the subject is taking a medicine, active ingredient or placebo. All these things, same color, same package, everything is same. But data management people knows it, which package contains active ingredient, which package doesn't have it. So that is known as single blinding, double blinding. These two are that concept of blinding, first of all. Okay. Now, somebody, can you please admit the students? They are in waiting room, please. Now, what is descriptive statistics and inferential statistics? Descriptive statistics is like, which summarizes the data using indexes such as mean or even standard deviations. Inference means we want to draw the conclusions. For that case, observational errors, sampling variations, etc. All these things. So, which is the best? For example, measures of central tendency. Everybody think about measures of central tendency, mean, median, mode, harmonic mean, geometric mean, etc., etc. Why we need this many of measures? Why we need this many of things? That we are going to discuss these things. Discuss to condense the data into a average. Suppose we have like hundreds of values, we want to take the average. That is one value. So then we want to compare those things. Per capita income of US, how much? Per capita income of India, how much? Now see statistics. Now India is the fourth largest GDP, for example. GDP. Recently they surpassed Germany. So that means if you consider about fourth, first one is America, second one is China, third one is Japan, and fourth one is India now. What is the per capita income? Per capita income, Luxembourg, $140,000. Simply, if you come to US, it may be like $48,000 or something. How much is India? Where India stands? Where India stands? India per capita income is definitely less than $3,000 or $4,000, this much. Number one is 120,000 Luxembourg, and now it is like a $4,000. That is the fact. That is the fact. So start, because when you see the definition, lies, damn license statistics, everybody is taking their own way to promote like this. I don't want to comment those things, but this is the fact. Per capita income is the important part for developing the growth of the nation. So you need to understand per capita income means all these 120 crores of people, they are taking them this much, and finally they'll get some number. Similarly in US also, similarly other countries also. So to condense the data into single value, to facilitate comparison between the data. See, if you understand previously what you are going to do, or what is mean? Mean is nothing but the total number of items divided by number of items, something like that. Now I want to change this one. Change this approach. Oh, we are going to use this one. There are two types of averages. We need to figure it out, like, every average has advantages and disadvantages. But we have like a five central tendency. Which one is ideal? Ideal measure of central tendency. Which one is ideal? For that case, we have some characteristics. Those characteristics are, it should rigidly defined. Most important, it should rigidly defined. Okay, characteristics of ideal measure or good measure of central tendency. It should be simple to understand and easy to calculate. It should be based on all values of the given data, all the values. It's further capable of mathematical treatment, sampling stability. It should not be unduly affected by extreme values. If you are taking about median, it is only influenced with only two values, middle values. If you are going with median, maximum repetitions, it's not depending upon all the values. Only mean is comes with here, is rigidly defined, simple to understand and easy to calculate, based on all the values of the given data. Further, if you want to calculate standard deviation, something like that, we are going to use mean. And sampling stability, it should not be affected by one value, it got increases. For example, if you know time series, moving averages are there. 3 years moving average, something like that. We are using, it is not at all affected with extreme values, because this is a link up. When you understand what is time series? A series of data in accordance with occurrence of time is nothing but time series. What is analysis of variance? Separation of variation into meaningful components. For that case, analysis of one-way classification, two-way classification, CRD, RBD, etc. Those things is different. So now you need to go with a bigger picture. Oh, statistics is wide, totally big picture. So we need to go with the step-by-step approach. So these are all any statistics measure which satisfies all these things, then I can consider that ideal measure of dispersion, ideal measure, allocation, mean, median, mode, harmonic mean, and geometric mean. So I told you, like, same thing like arithmetic mean, it is rigidly defined. It should be simple, and based on all observations. Further mathematical treatment.
Sir, we are unable to hear your voice, sir.
You are in mute, sir.
Hello, are you able to hear me now?
Yes, good.
Okay, perfect. Thank you. So now going back, like, so mean and standard, if you want to work with the demo data set or adversely, because why I want to throw all these words is, like, if you want to enter into corporate, you need to know a lot of things. For that case, select the right person first. That's what exactly we put a Chanukya's emblem here is, select the right guru first. If you are at right guru, you never did anything wrong. If you are not at right guru, whatever you are doing is wrong. So first of all, you need to get a right guru first. Most important is, now going back here. So arithmetic mean is the best for all averages. It's almost all the requirements for ideal measure, all these things. Next, measures of dispersion. How data is going to be deviated? Suppose a person having with 90 marks in one subject and 90 in another subject, third subject is 0. 0. The average is 90 plus 90 plus 0, that is 180. 3 subjects by 3, average is 60. Another person got a job, marks in 62, 58 and 60. Average is 60. Both having the same performance? No, absolutely no. So on that time mean fails. Then what we need to do? How data is deviating? How data is deviating? Xi minus x bar whole square. That means average is 60, 90 minus 60 whole square, and again 90 minus 60 whole square, and 0 minus 60 whole square. We are taking that. This deviation is more. He is not consistent. He is not consistent. So that means certain situations mean fails. Then we are going with the dispersion, standard deviation. Again, here also, range, quartile deviation, mean deviation, standard deviation. Range is only depending upon two values, two extreme values. Quartile deviation, Q3 minus Q1 something. That means Q3 is 75 and minus only 50 records, 50% of the records. Mean deviation, modulus of something. If it is negative, comes into positive values, same thing. But only standard deviation go with further levels. So that is the reason all this first leg we want to calculate mean and standard deviation. Because of this reason, those two are the ideal measures for dispersion as well as location. Okay, and now somebody is asking a question: how to calculate sample size? Is there any particular software? There are so many tons of softwares are there for calculating those type of things. Any one of you, can you please, if you want to put a question, please put your name. Of course, name is already your educational background and question. That should be better because you are belongs to medical background or pharmacy or statistics. Then it should be it's more appropriate for me to explain in a better way actually. Okay, please. Now, going back to here, I must based on our observation of the data, etc. That's what I'm explaining. Variance is equal to square of standard deviation. Now, key statistical techniques. What are the statistical techniques we are applying in clinical trials? When you are applying the statistical techniques in clinical trials, very first one is randomization. Random ensures unbiased allocation of participants to treatment groups. You need to understand what is unbiased, what is allocation, and what is treatment group. What is treatment? How we want to make it a group? And it is an open label study or crossover study. Because why I am going to use these words? Because you need to understand, if you want to get a job, you need to learn a lot of things. That's one reason. First, blinding reduces the bias by preventing participants and or researchers from knowing treatment assignments. That's what I clearly mentioned is blinding. Sample size calculation. I told you in future slides, I explained about how to calculate the sample size. That's very, very important. Sample size is very important. Suppose, for example, we have like 100 people in this conference. Almost a lot of people watching in YouTube also, because we have 100 licenses. If the person is more than 100 licenses, they are going to open YouTube or some other places, something like sample size calculation. Hypothesis testing. What is hypothesis? What is null hypothesis? What is null hypothesis? Everybody knows about it. What is null hypothesis? The neutral or non-committal attitude of a statistician is nothing but null hypothesis. The neutral or non-committal attitude of a statistician is nothing but null hypothesis. Are you able to hear me?
Yes, sir.
Okay. Yeah, somebody put it like you are muted or something like that. Okay, anyhow. So how we want to assign the null hypothesis, how we want to go with the next level. So that those type of things, like it's important. Confidence intervals is 95%, 99%, something like that. So then after the survival analysis, evaluate time to event outcome such as disease progression. This is how diseases progress. Survival is very, very important part. Regression models, relationship between two variables, y is equal to mx plus c or y is equal to a plus bx, something like that. Relationship between correlation is established relationships, but here is you need to go with a positive relation or negative relation. But which variable is dependent on which variable? That is regression comes into the picture. Supply and demand. Supply is depending upon demand. If demand increases, supply increases. If demand decreases, supply decreases. So demand, supply is, if supply is increases, demand increases? No. That means y on x is not equal to x on y. It's commutative property. A into B is equal to B into A. That is commutative property. Same commutative property is not applicable in regression, because the regression for A x on y is not equal to y on x. Simply same thing. There is relationship between supply and demand. Supply and demand. So if demand increases, supply increases. Supply increases. So demand is depending upon the supply, but supply is not at all depending upon this, like this. So that means that is the reason you need to understand the regression concepts also. Now, also interim analysis, Bayesian models, inferences, etc. Everything. These are all the very, very important. Tomorrow, when you are going to the corporate, yes, these are all the important. And of course, adjustments is a little bit required. Principles of experimentation. Everybody, because we are conducting so many experiments, principles of experimentation is everybody knows about. If you have a statistics background, that is like replication, randomization, local control. Replication, randomization, local control. Replication means repetition of treatments under investigation. Randomization, you know that, like, local control, the process of reducing the experimental error, something like that. See this one, randomly assign treatments to experimental units. Replication, repeated the experiments under investigation. Local control, minimize the local errors, local variation, blocking and balancing. So these are all the major principles in experimentation. Suppose when you are going with your study, probably you will see like replication is the number one, and randomization and local control. Of course, blocking and balancing is also very, very important for this much. Okay, these are all the things comes into the picture. Now, when you are going with a biological experiment, always think about like, biological experiments is like biology, pharma, agriculture, and life sciences. Also like purpose. First of all, when you are conducting experiment, what is the purpose of this experiment? What is the scope, objectives, objective hypothesis, methodology, conclusion, practical applicability of the proposed research. This is the very important. When you are doing a research, first you need to decide, hey, what is the purpose of this research? What is the scope? Where it is going to be applicable? Where it is going to be used? Those are all things is very important. Very important. If we have answers for all these things, definitely our research is good. Research is good. Otherwise, it's different. Okay. Now, so analyze the data is the more important. Researchers contact statistician only to analyze the data, but this involvement may be too late or cannot compensate the poor planning and collection of the improper data. That is going to be happen a lot of times. Simply like, that's the reason. That is the reason people like me, I am working in end-to-end. End-to-end means in a clinical trial process, I'm giving a whole structure. The structure is draft protocol approved, the first step. Protocol approved. Contract non-disclosure. Informed consent. First subject, first visit. Recruitment starts. Last subject, first visit. Recruitment completed. And last subject, last visit. Treatment completed. Then database lock. Stat analysis ready. CSR final. This is the whole process. When you are conducting an experiment, you need to understand all these things. Simply after some time, we are collecting the data, and the data goes to a particular statistician. Oh, I want to analyze the data. So if the data is not in right direction, we are not able to analyze the data. So first of all, when you are planning a research study, such as agriculture scientist, medical doctors, dentist, and entomologist, all these things, perfect planning is required. Perfect planning. For that case, lot of statistics. So much of statistical methodologies is available here. For example, define the problem you intended to study. Rate of oxygen consumption with thyroid T4 by injection. Rising trends in AIDS, smoking and cancer, radiation and leukemia. First of all, identify the problem, how this one. Then you can add one more thing also, how social media affecting the student behavior. Something like that, something like that. So relationship, that is my problem. That is problem I intended to study. For that case, you know about all these things, smoking and cancer, for example, you have the people who had suffered with cancer and smoke how many cigarettes. Then you need to establish relationship. Oh, smoke, if you are making more smoking, and the probably chances of getting cancer is high. Something like that. If you don't have, let's simply you are collecting the data, and after that I want to see the problem, it doesn't work. It doesn't work, first of all. So make sure that, make sure that you need to consider about first of all, define your problem which you intended to study. That is the first step. Second step is what is your aim? What is your objectives? Then, state whether solution has to be found by different methods or by nature of the problem. State it. For example, modify measures of controls enhance inflammation of the brain is rising. People having a smoking habit is one of the major factors of lung cancer. Lung cancer. That study is there. The research is there. First of all, you want to analyze or you want to come up with another conclusion, then probably it may be yes. So define the aim and objectives of the study. Then after that, literature review. The most important is reading the literature. See, dissolved oxygen in water is required for fish respiration. But one wants to know minimum level of dissolved oxygen in the water of a survival fish. But I want to know about, I know that, definitely, that dissolved oxygen is required. How much is required? Yes, for that case, you need to do some analysis. And hypothesis. B complex injection is not more effective than by mouth. See, B complex injection is not more effective by mouth. Is it a null hypothesis? No. We want to make sure that null hypothesis means there is no significant difference between taking the B complex injection by mouth or by injection intravenous. Something that is my null hypothesis. Then my null hypothesis is wrong. For example, that means some difference is there. Then I want to go with alternative hypothesis, left tail or right tail, whatever. We'll go with next level. So hypothesis is very, very important. Null hypothesis and alternative hypothesis. Once up to that part, then up to that part is done, then selection of sample, specifying the nature of the study, rule out by the instrumental error. When you are taking some instrument measurements, you need to avoid the error, first of all. That's very, very important. A lot of times, lab information, they put it in the wrong settings. That's really big mess, first of all. Reading the data in a standard format, schedule, perform, etc. Suppose when you are working with big companies, they are all going with ISO 8601 format. ISO 8601 format. Across the globe, if you are in pharmaceutical company, you need to follow that format. They need to follow some standards. Why they want to put standards? For example, 0506 2025. If it is in the US, I am going to consider that is May 05 is May 6th. Suppose that same data is in India, they are going to consider June 5, something like that. So that means everybody should follow certain things in a different way. That means we need to follow those things. Suppose, for example, if I am asking a question like what is probability? Suppose somebody mentioned M by N, for example. What is M by N? Probability means across the globe, you need to mention that total number of favorable cases divided by total number of exhaustive cases. That is important. If you are going with Boyle's law, PV is equal to constant. What is PV? Is not a PV not tomorrow. PV, but the definition is pressure is always inversely proportional to volume provided temperature remains constant. That is the approach is required. So you think, learn locally, expose globally. That's very important. I don't want to say if you go to some Kapur Gupta book, they are going to mention M by N or something like that. That is fine if you want, but if you come down globally, but you need to come up with what is M, what is N. You need to explain them. Oh, total number of this by this, something like this. That is more important. That is the reason thumb rules. We put it like join with your first name and last name. See, second one, I saw like somebody joined with 919-819-737-2007. What does it mean? Is it his name? No. No. You please understand, like, just to change the name. That is the standards. And if I am taking the classes, I want to activate your videos. Because why I want to activate the videos? I'm able to see your face, whether you are able to understand it or not able to understand. Understand, that's the one reason. All the meetings nowadays is Teams or Zoom or video conference. So that means that's very, very important. And the next step is like, when you go to the next level is like, you need to come up with your concrete questions. That's the more important case. Okay. Now, every precaution should be taken to avoid error because correct reading will give correct statistical analysis to draw the correct conclusion. Correct conclusion. Instruments like weighing machines, height measure, okay, hemiometer, respiratory meter, all these things should checked before when you are recording the data. When you are recording the data, all these things is required. Presentation. What is then once it is done, somebody need to know about it. We want to present the data. How we want to present the data? There are so many technologies are there. So many technologies. Lot of times, as a clinical trials analyst, like SAS programming for so many years, my major responsibility is I'm using that word. My major responsibility in my work is creating the TFL. TFL means tables, figures, and the listings. Tables, figures, and the listings. Sure. So in these tables and listings, figures means like a diagrams, for example, graphs, for example. Okay, now what is a table and listing? What is the difference between table and listing? When I am conducting an interview, I am asking a simple questions. What is function? What is function? And what is the difference between table and listing? The people, they don't know these things. Yesterday in Hyderabad, the US consulate, they conducted a technical screening for F1 student. A person completed data science degree in India. That American officer, immigration officer asked a question, what is dimension? First question. And what is array? Second question. You are a data science student. Can you please tell me what is linear regression? Only these three basic simple questions he asked it. Unfortunately, he put a statement, I surprised with the student performance. Sorry to say, that's the fact. So basic things, we don't want to go with what is the difference between table and listing, or what is function, something like that, okay? Now, going back here. So unbiased statistical analysis applied by different methods in biostatistics. We can put all those things. That's not a big problem here itself. See, when you are going with these things, these are all the steady problems, collection of data and compilation, analysis of data, all these things. Like methods, I told you, like, there are so many softwares are available in the market, Of course, I am working with the SAS software for so many years. Now I am moving towards a little bit about like R software. Why I am working with SAS side? 95% of Fortune 500 companies, they are using SAS. 100% pharma biotechnology companies, they are using SAS. That contains money. Actually, frankly speaking, money is the very good motivating factor for every person. By end up, why you joined in this session? You want to learn something. That's all it is. You want to learn something. Same thing like that. Same thing. So we are getting a good salary for this. Definitely, one software is what is sample? Yes, this is the sample. This is the population of interest. I don't want to go with conducting experiment with the whole population. So that's the reason I took a sample of like 5 or 6 people, depending upon that. Okay, this is a sample is relatively a small group of selected number of individuals or objects or cases drawn from a particular population and use it to throw light on the population characteristics. That is the reason population means a set of individuals having common characteristic is a population, and sample means a finite subset of population is nothing but sample. A finite subset of population is nothing but sample. So that means you need to understand what is exactly population, what is exactly sample, what is population mean, what is sample mean. Expectation of small x bar is equal to capital X bar, something like that. So these are all the things, apply those things. Random sample, randomly I am taking like this. Each and every individual has equal chance to enter into sample. For that case, if you have like 100 people, I want to take it only 5. What I need to do? I'm taking 100 names into a piece of paper, same color, same folding, everything is same. I can put in a on and shuffle it, and I'm taking only 4 or 5 what is required. Then are you going to consider I am a bias? No. Is it like randomization? Just like a random sample. That type of things is required. Next, of course, sampling. See, for example, if a biological experiment is rarely possible to collect complete information about a population. Impossible, first of all. We cannot take out blood of the whole body if all the individuals have population of HB, V1C or RBC count. What we are going to do? A small pinch of blood, and we are going to apply those things. Whatever, lab results apply to whole body. This is what exactly sampling means. And somebody asking about like sample and population, etc., etc., all these things, like, all these things. No, coming back to here, a finite subset of population is nothing but sample, something like this. Essentials. No, merits and demerits, of course, these are all the things, like, so many information is there itself. Like I told you, like, this is a sample size. There are so many ways, correlational studies, descriptive studies, etc. Somebody asking like a calculation of sample size with the confidence limit is equal to 95%. This is the formula. You can apply this one. N is equal to capital N divided by 1 plus something like this is the formula. We are going to take it entrance level. Then you are taking about, if you want to take it 95% of confidence interval, at least you need to have 10 people in a sample. 10 people. This is the way you can calculate this sample size. Somebody asked me that question. This is the way you need to calculate the sample size. Okay. Of course, if the number is equal to 10, sample size is 10. Number is equal to 50, sample size is something 44. Number is equal to like 100,000, your sample is 384. This is the way they calculated the sample size. These are all the general rules for calculating the sample. General rules for calculating the sample. That's what we want to do this like this. There are so many ways of this sampling is there. Populations of simple random sampling and stratified random sampling, cluster sampling. So many methods are there for sampling. Because I told you, like, this is within 1 hour or 1.5 hour session. It's impossible to explain each and everything, I'm sure. But it takes a little bit of time. But you need to understand a lot of things is required to learn to get into corporate. That is the one thing. It's like, the sample method, table, random numbers. These are all the numbers. This is the way we want to take it, like random numbers methods, random numbers, etc. Everything here. This is the systematic sampling. Cluster sampling. Suppose statistical methods. We have a right problem is here. Psoriasis is a chronic disease of the skin characterized by a variable clinical failures. A clinical study is conducted to evaluate the effect of an experimental drug in patients with psoriasis disease. This is the problem. That's what I gave the 2 pictures like this, because you need to come up with this. First of all, how we want to do this? For that case, design. I want to design my study. The study is randomized, parallel group, placebo controlled trial in a total of 112 subjects. 112 subjects will be randomized with 1:1 ratio in 2 arms, 150 mg test drug and placebo. How many of you understand this one? How many of you understand this one? So, because that's if you conduct a study, that study must be randomized, parallel group, placebo doesn't have any active ingredient, controlled group. This is the approach. Then you want to take like 112 subjects with 1:1 ratio, 1:1 ratio. The primary endpoint is the level of decrease in the baseline psoriasis area and severity index so and so in 12 weeks. It's not like one day or 2 days. It's like a 12 weeks of this, No, like this. So when you have these type of things, then you need to develop contents. What is required? Your subject ID is required, subject identifier, and BPASI, baseline information, and BIGAN, baseline with the 1GA, etc. Everything, height, weight, etc. All these things is required. For that case, what I am going to use. I am going to use a SAS software using proc contents. Data is equal to so and so where num. When I am going to write these 2 lines of the code, just these 2 lines of the code, then you will see all these contents. Hey, my data set contains this variable, this, etc. Everything there itself. Now, what is your objective? What is your assumption? And of course, X bar, sample standard deviation, your null hypothesis. And depending upon the study, I'm going to use this test. That's the statistic. So this is the approach, like you need to do this. Then you can do the SAS code for proc univariate data is equal to so and so, etc. Everything. Then you are applying all these things. And finally, you got the reports like this. Then statistician is going to think about it. He wants to come up with a final decision. Oh, whether whether it's really effective or not effective, is a significant difference or not significant difference, whatever it is. Something we can do this. So your role is, suppose if you are a pharmacy background, your role is you are going to create a program and run this part. Proc univariate data is equal to so and so, and MU this much normal and plot so and so, etc. This part is enough. Then statistician is getting the results like this. Then he is going to take a decision. He is going to take a decision whether we want to go further or not. We want to take it those things. So this is like, this is the approach. If you go with like this, see all these things, F test or T test, depending upon situation, which situation we are going to use T test, in which situation we are going to use F test, which situation we are going to use chi-square test. All these things you need to decide, because certain conditions we need to apply it. We can get all those things, T test procedures, etc. Everything here. Oh, these are all like software based. Even if you are going with R software or even stat like S plus or S is also there itself, S also, something like this. These are all the things you need to figure it out. You need to understand these things, apply those methodologies. Methodology, you are able to get it. Risk and risk differences in SAS, etc. Everything. Don't worry about that part. If you want to, because a lot of times if people are calling me, I want to get a job. I want to get a job means tell me your skill set. Tell me your skill set. What you know that? Oh, I'm getting 80%. I don't care about 80%. Just like tell me what your understanding. What are your understanding? So that type of things is required nowadays in a corporate. So something like this, testing methods, lot of things we can get it. We can get these results. Certain things, you can get it. Something like this. When you go back to here, suppose NPARWay is another procedure we are using with SAS side. Okay, here also to test the treatments difference in change from baseline, so and so, etc. So certain things. First of all, you need to have a objective of your study. How you want to reach that objective. For that case, you need to write what type of data is required, what type of statistical methods is required. What type of conclusions we are going to do this, what type of hypothesis. These are all the things, step by step process we want to do this. And also like we want to identify the type 1 error and type 2 error and level of significance, power of the test. All those things is required to get in this much of a level. Generally, these are all the things. Proc means is to calculate descriptive statistics. Univariate is for sample t-test. T-test is for difference between two samples, mean differences. Proc frequencies, n-power way, etc. These are all like summaries, etc. Everything. There are generally when we are going with I'm giving a task like this. A background hypertension is a chronic medical condition which the blood pressure in arteries is elevated. The clinical trial was conducted to elevate the efficacy of the experimental drug A, anti-hypertension, AHTN. For that case, I want to conduct a study like this. A randomized parallel groups study conducted. 75 patients were randomized in equal numbers in three treatment groups, high dose and low dose and a placebo. So if I am going to do these things, a statistician is going to write a analysis plan. So he wants to analyze the data, how you want to do this. He wants to what type of data is required, what is the primary endpoint, what is the secondary endpoint, what type of statistical tests are required, all these things. In the whole situation, I never talk about what is t-test, what is f-test, what is binomial distribution, what is normal distribution. I never ask all those things. Everything is available if you have a smartphone in your hand. Everything is available, but you need to know in which situation which is appropriate test. You need to justify it. You need to justify to FDA, hey, this is the reason I am using this test. I am using this test. So that is the more important, you can do these things. Similarly for data, see PAT, ID, treatment, etc., everything here is something like this. These are all the things you need to go with. Then check the baseline blood pressure are comparable across the treatment groups in pairwise fashion. Question 2 and question 3, they are asking so many questions. You need to answer those things, this much. This is the one important approach. And people really wants to come to like a SAS programming or the status this side. Think about this type of certificate, SAS certified advanced programmer or something like that. Even this is the base programming. And if your resume has symbols like this, I am a base SAS certified. If you see like this is Venkati Kurti, and I got the certificate almost 2008, same day both certifications. So that means that once you have a certificate, I'm very positive you will get a interview. You will get a interview. I'm not giving you will get a job. I'm very positive you are able to get an interview. You can talk nicely. Definitely you will get a job. Now this is a good time, particularly in India, because BMS and Novartis and Bayer, Sanofi, Amgen, all these pharma companies, they started their offices in Hyderabad or Bangalore. So there are plenty of positions are available, exclusive have people with these skills. I told you like 5 skills. Those skills have it 100%. You are getting to the market with a very decent salary, starting with at least 7 to 8 lakhs starting salary. You are able to get it, and within a couple of years you have a better. I have so many students, they started with like 4 to 5 lakhs. Now they're all making with 30 to 40 lakhs. And in somebody in US, somebody in Germany, somebody some other places. So there is, if you have a skill, nobody is going to stop you. If you don't have skill, nobody is going to help you. These are all the symbols you are able to get it. And this is the feedback form. You can scan it or send a feedback form, like scan, and you can give the feedback form. That should be good. And of course, this is also another feedback form. We can do this. So this much. And somebody, this is now that this is the question and answer session. And if you have any questions, so definitely we are going to address these questions one after another. Okay. Now going back, somebody is asking a question: how to get a sample size for these kind of studies, and what I have to take in primarily a primary endpoint. So, Raman, can you please unmute it? And what is your education background?
Hello, am I audible?
Yes, go ahead.
Hi, good evening. So I am MSc Statistics,
Okay, which university?
University of Lucknow.
University of?
Lucknow.
Oh, Lucknow. Oh, nice. Okay. Because that is long discussion, frankly speaking, depending upon the study primary endpoint, and because investigators, all the people are going to involve for endpoints, etc. Then after that, send me the small note, we will discuss those things. Yes, definitely we can do this. Is long, because a lot of people with a non-statistics background, that is the reason they don't want to upset you. Definitely I can provide you some good information for this, Raman. And
Got it, got it.
how can statistical methods be used to access generability of clinical trial results in a wider population? Clinical trial results in a wider population, when you are going with this, preclinical studies, phase 1, phase 2, phase 3. Phase 1 is 10 to 30 people. Phase 2 is phase 2 is like 100 to 300 people. Phase 3 is 1,000 to 3,000 people. That is the generally we are going to standard. We are going to conduct the experiments with this, first of all. So if you see like this, if it is wider population, is only they are conducting this information in phase 4 only. Phase 4 is a continuous process, that one. But getting the approval up to phase 3, and these 3,000 plus 300 plus 30, around maximum 4,000 people, we are conducting these experiments. And what is some other mentioned, like what is interim analysis? Interim analysis is because clinical trials is a very expensive thing. If we want to go further or not further, then we want to conduct a analysis in the middle. Definitely we can, depending upon that, we want to go with that same type of reports we are going to generate. The reports are positive or negative. That's the reason people call it as interim analysis. Any other questions?
Yes, sir. In chat window, we have some questions, sir.
Go ahead. You can type the questions in a chat window. Oh,
How
okay.
can
What are the-
statistical methods? Yes.
One second. First of all, feedback form, and then after the calculation of sample size, just now I discussed about it. And sir, are you providing any SAS certification program? How many months it will take? SAS certification training and everything is yes. We are going to provide you the trainings for this. Most of our trainings are in the US and India, online and offline also. Oh yeah, you can get it See, again, I'm telling the why you want to learn SAS. Suppose, for example, somebody asks you to Bindu, Upendra, something like that. Why you want learn SAS? Probably they want to think about, I want to get a job. Getting a job is learning that particular subject is not that much useful. That is the reason we put like a 6 months internship program. Internship program means in this internship program is they are able to understand basic statistics. I mean applications of statistics, basic application of statistics. Then after that, you need to work with some dummy data. It may be hemodialysis study or something like that. You need to understand protocol and CRF and annotated CRF and C-disk standards, converting these tables, figures, and listings, all these things. So definitely it will take at least 6 months to get in that level. Definitely on that time, what happens is a lot of my students working in Novartis and other big companies, because the 6 months is ideal. Suppose if you want to learn, definitely we will provide you the training. Absolutely, you can contact us by email, or of course you have our phone numbers. Then you can contact us. Absolutely, it's not a problem. Software for calculation of sample size N. Sample size calculations, even you can use with SAS software, also you can use with the sample size calculations. You can use even R also for calculation, and prepare with the SAP. I suppose you did not mention your name actually. That's a number. I told you if you want to type it your name and which university and what is your education background, then that should be better actually. That's the one thing. Yes, there are so many softwares are available to calculating this sample size. And of course, preparing the analysis is different. Okay. Any other questions? One minute.
In the chat window, we have another question.
Okay. And I am doing my master's in statistics. Now I am unable to find a job on my education, related education. That's problem is I told you, like, there is a huge gap between industry and education. Industry and education, we want to minimize the gap. That's very important. Further case, internships is going to reduce a gap between industry and education. Coming back to statistics, you are talking about statistics. Tell me, in industry, how you are going to use your statistics, how you are going to apply your statistics knowledge. That is the very important part, very important part. If you don't know, just simply learning mean is equal to this much and mode is equal to this much, something like that, is really tough to get a job, now the industry is looking for quality people. Quality means they are ready to take challenges. They are ready to take challenges. They are ready to learn the subject. I don't know which university you are from. Basically, I am from, like, Nagas University. On that time, when I am studying in Nagas University, I don't know even word of the SAS, first of all. SAS. Then I came to US. I worked as a lecturer for almost 10 years. Then I moved into IT side. Then I came here. Then I'm doing something. I learned my work. Then I am going to educate the people to get all these things. Yeah. Yeah, this workshop is not clear about anything. That's what is exact. This because the reason why I put this workshop is like, as a statistics person, learn lot of things to get into corporate. A pharmacy student must learn these things. If a person goes to like a corporate, you need to go with like clinical trials, drug development, regulatory, all these things is required. Suppose, pharmacy student, after 2002, a pharmacy graduate from India is not eligible to take NAPLEX examination. So that means if they want to enter into the corporate, they need to learn these programming concepts. That's one thing. A statistics person wants to enter into the industry, a quality control, statistics people knows about all this quality part, but mechanical engineering going to get the opportunities for these positions. So that's the reason we want to prove that ourselves is more good. That is the main intention. This session is exclusively for people already in industry. That's different. People wants to enter into the industry. That is the one thing. Somebody mentioned I'm working as a professor, I'm doing something like that. This session is not exclusively for them, for sure. But here is like this. And also I'm giving a very high level approach. That means you need to understand a lot of opportunities, learn lot of things. That is the main intention of this presentation. In this presentation, don't expect like everything is to be explained within a 10. If I'm going to explain like mean, median, mode, harmonic, geometric mean for 1 hour, probably you're all happy, but doesn't help anything. Help is like, okay, these are all the skills required to get into corporate. That is the main intention is. Intent to treat ITT population. I am okay. Intent to treat and per protocol population is in protocol. They are going to clearly mention a flag, safety flag, ITT flag. Some flags are there. Depending upon that, intent to treat. So we are taking the patient's intent to treat. How many are there? That is the reason intent. There are different flags. When you are working with the CDISC standards, you are going to apply all those flags. Okay. Any other questions, Swapna?
Sir,
Yep.
in chat window, any resource suggestion to read about the sample size for a clinical trial?
1 second. Sudhakar joined the session.
Yes, sir.
Where his name is?
Vardhan, sir. Vardhan.
Okay.
Good evening, sir. I am very much here, sir.
Oh, thank you. Thanks very much. Now this is the time you can give our closing remarks. Yep.
Yeah, I'm very much here, Andy. Yeah.
Oh, good.
Am I visible, Andy?
Yep. Yes, sir. Go ahead.
Yeah. Yeah. Shall I go ahead, Andy?
Yes, sir. 1 second, sir. Kaviti, sir, is on the call also here.
Yeah. సార్ను ముందు మాట్లాడనివ్వండి, తర్వాత నేను మాట్లాడతాను.
Okay, sure.
అంటే మనం 8కి క్లోజ్ చేయాలి కదా.
Sir,
Venkat garu, it's a wonderful session actually. Even as a non-statistic students, it's excellent.
1 second, sir. Sir, all of you students, sir is like our international advocate, and he has offices in all 26 countries, and is well prominent attorney, helping thousands of students, particularly in US, because if they have any legal issues or something like. So Shar is sharing some information exclusively for the people, students who wants to come to abroad, particularly for who wants to come to abroad. These are all the precautions. And also, we both are in of course, even Sudhakar also in the same boat. If we have skills, nobody is going to stop you. If we don't have skill, nobody is going to help you. Sir, go ahead, sir. Now this is the time for you to explain your advice for these young kids.
Yeah, first of all, Venkat garu, it's an amazing service what you're doing, and on a Sunday, you are giving your time to people all over the world to enrich and enlighten with your wisdom,
Sir,
and
if
it's
you want, you
commendable.
can activate your camera, sir. It should be better actually. Everybody is able to
Yeah.
see. Of course, you are a very popular person.
No, sir, it's okay. So that's what it is. I would say, because you're doing such a service, they all have to take advantage of it. The other thing is the in terms of students, whether it is in America or in India, if they want to get an employment, because I also have many companies and I hire people in India and UK and America. The first thing, as you said, it's not the education is the key, but it's of course is the skill. And additional with education, if you have the skill, you have employability factors high. And if anybody wants to really come abroad in terms of US or UK or Canada or Australia, I'm sure these days it's a prestige, as well as they can make better money than India. As you said, starting salary would be 7 lakhs or 10 lakhs in India, and they would make, I would say, 60 to 70 lakhs starting in US or in England or any other parts of the world if they have skills and education. Of course, they have to be competent. And we have been helping, and in the past, I have hired myself SAS programmers. In the past, I paid them 70 to 80,000, even $100,000 salaries. I sponsored them from my IT consulting company in the past, so I'm quite aware of it. And I did sponsor some clinical research associates in my company. I had a medical consulting company. I mean, they were paid 75 to $80,000 starting salary. So what I mean to say is, as you rightly said, one has to have a good education, of course, and skill. And the third factor is the communication. Many of us, when I see in India, they have all gold medalists, but when they talk in English, they're doing grammar mistakes. So when they're writing, they're doing grammar mistakes. I mean, of course, you have a knowledge and skill, but if you don't know how to communicate verbally in a spoken English or in a written English, people will judge you based on your or written and oral communication, and that is the key. And the reason I am able to share, Venkat Garu, because I have a master's in communication. So communication is the key. I might have a law degrees from many countries, but if I don't know how to communicate and if I don't know how to write, that's the poor thing. So what I suggest all the students that they should take classes like this, and take the knowledge from individuals like you, because you're doing a lot of seminars and workshops for students and as well as professionals. And as we spoke in the past, that we are here to assist and help people, those who want to migrate to America or any parts of the world, with knowledge, skill, and employment, and definitely in terms of guiding them in terms of how to get a visa, how to settle. The other thing I wanted to share with your group, the lot of PhD guys can also migrate to America or Australia or Canada with a PhD degree with an extraordinary ability. They can immigrate with a green card. So those also we can help, in terms of any of your students or any of your clients, those who intend to migrate to other parts of the world. I guess I take this opportunity to introduce me to your group, and we would like to do more and more with Chanukya Educational Hub. And as Venkat itself is, both the daughters are very successful doctors, and himself being a statistician and a statistical programmer and data scientist, and being a very famous lecturer in Andhra. So I guess we should thank him. And I really deeply thank you on this Sunday, you taking out time and doing such type of workshop. Somebody said they don't understand the purpose of this workshop. Well, they might be able to understand the purpose of yours in 1 hour session. But if they keep following you and getting in touch with you, I'm sure they will accomplish their purpose. And thank you very much once again.
Thank you, sir. Thanks very much. Yeah, sir just now mentioned about yeah, definitely EB1 is for extraordinary category, people having with the doctorate degrees. There is a special provision for, applying the green card and everything. And sir has 100% success in that particular areas. If anybody wants, you can communicate with us, definitely we will give a contact information to sir. Definitely he will help all these things. Now, this is a term for Sudhakar Garu. Sudhakar Garu is a Group 1 officer in AP state government. He's recently got retired, and he's a technology evangelist. He's a Prince certified, etc. Continuous learning is the key for the success. So that is the reason I invited him to talk something about all these things. Sir, go ahead. This is your term.
Yeah, yes, Andy. Am I visible and am I audible, Andy?
Yes, sir. Go ahead, sir. Yes.
Right, Andy. Thank you very much, Andy. And one thing, ఏంటంటే, I mean,
Most
I
of the
always,
are North India also, sir.
am I audible and am I visible, Andy?
Visual and audible, please talk in only in English,
Yes,
sir.
sir.
Because a lot of people from out of Andhra Pradesh.
Right. Fine, Andy. That's fine.
Okay.
See, I always tell people that the very first interaction with Dr. Venkati Kurti was when I took my daughter to join the course, the course she was giving for SAS. So my daughter passed both of them. She worked for some time. Now she is in Canada. Okay, that being said, see, a lot of people were expressing their own doubts regarding their learning, whether they're able to understand or not. See, this is only an introductory session where he has given you a small guidance kind of a thing, right? See, you should know that Dr. Venkat Kurti has 25 years of experience in statistics and as a data scientist. And whatever he said, it's palpable and it's visible in every sentence he said. The slides contain not only knowledge but ample application pointers. See, this is what you all need to understand. See, a lot of people were bothering about certifications. See, this certificate this is only an introductory session. You will not need a certificate for an introductory session. Okay. So now, having said that, I want to inform you that one thing. One sec. Yeah. So now, Dr. Venkat's, the experience, the exposure is very, very high, and he has not only been a data scientist and a statistician, but he has been training. See, training and there is theory and practice. See, not everybody has both the theory and practice. So he has a lot of theory and practice. Now, what I want to tell all of you is, after having listened to this enlightening session, you all need to understand in your own wisdom the usefulness of this internship of 6 months in statistical methodology in clinical trials and drug development through SAS application. See, SAS application is not a glorified Excel. It's not that. It's much, much more than that. And as he has already said, and already as most of you know, most of the Fortune 500 companies, almost all the Fortune 500 companies use SAS methodology for clinical trials and drug development. It is a given. Now, all of you have done your courses in statistics, and now you are very good in theory, right? But now you need to ensure that you apply what you have learned. And this course, internship course of 6 months of this statistical methodology in clinical trials and development is your passport to success. So, Mr. Venkat, Dr. Venkat Kurti would be helping you on this training journey, and I am sure that having known him for the past 15 years, he is a lifelong learner, and he was telling that, probably that is where we gel together, we vibe. So my request to all of you is, now that hopefully you have taken notes on what Dr. Venkat has propounded today. If you have not taken notes, then it becomes difficult for you. You have to take small notes so that you will understand what is the the learning that he will be giving it to you. Sir, I take this opportunity to thank you and your team for making this session a very lively and interesting session. And I also thank Dr. Srinivas Garu.
Yep, Srinivas. Yes. Yes, sir.
Dr. Srinivas Garu, Srinivas Vemuri Garu kadandi.
Kaveti.
Dr. Srinivas Kaveti Garu for being here and talking about his experiences and how you also need to do. And thank you all the participants. So thank you for giving me the opportunity to give a vote of thanks. And it's a privilege, a pride and privilege for me. Thank you, sir. And one more thing. Dr. Srinivas Kaveti Garu was concentrating on the power of communication, right? So now, as a tech evangelist, I would also like to tell you that, you also will need to see, once you learn this, see, for example, a semicolon, missing a semicolon will lead to chaos in the modeling that you will be doing, right? So you are referring to only a semicolon, right? You miss a semicolon, the entire modeling goes wrong. So that is the importance of application of SAS in this clinical trials and drug development. So here, as Dr. Srinivas Kaveti Garu has aptly said that, please ensure that your communication skills are also developed along with this, because sir also what he does is in all his courses, he also adds a lot of, soft skill development to as a bonus to all the learners, and the communication skills part and project management, all these would be part of that bonuses. So thank you very much, sir. I mean, it's a pride and privilege to be part of this session, sir. Thank you.
Finally, I can say only one word. Chanika Education Hub is a single source for all your education needs. That means a single stop. A person wants to come to abroad, we will help some extent. If we want to help the job, if you have any legal problems, sorry, it's here. And of course, I'm in US, like this is a single source. You need to have a authentic information. That's more important. A lot of times people, they don't have authentic and reliable information. If you want any that type of information, please contact us. Definitely, we will help. Thank you. Thanks very much, all of you, for joining this session. I know your time is already 8 o'clock. We're on time for sure. 8 o'clock now. Thank you. Thanks very much. Sapna, I'm leaving now. I can make you as a host. Okay, then, right?
Okay, sir.
Okay, good.
Right, sir. Bye, sir. I'll take leave. Thank you.